Hello, Miss Hain. I will explain about evolution. Basically, in general, evolution is changes in population, species, or groups of species. It is the process where frequency of heritable traits in a population changes from one generation to another generation. So the genetic variation in a population is determined by mutation, sexual reproduction, natural selection, genetic drift and gene flow, microevolution, and macroevolution. There are a lot of scientific evidences that support evolution. The first evidence is scientists have acknowledged direct observation of evolutionary change. So the first example of this would be the change in beak size and shape in Galapagos finches after a severe drought. The second example is the change in predation and coloration of guppies after the change in the predator. The third example is rare resistant viruses had spread quickly when each of the patients was treated with the anti-HIV drug 3TC, and within a few weeks, 3TC resistant organism made up 100% of the virus population of each case. The second evidence is the fossil record. The fossil record shows past organisms differed from the present organisms, and it also shows that many species have been extinct. Fossils also shows the evolutionary changes that occurred over a long period of time. The third evidence of evolution is homology. Homologies are body parts that are similar to one another in different species because they have evolved from a common ancestor. The fourth evidence of evolution is biogeography. Scientists have predicted that the continents were connected long time ago. However, the continental drift had distributed the land. And the fifth evidence of evolution is molecular biology, and this researches the nucleotide and amino acid sequences of DNA and proteins from different species. Similar species share higher percentages of sequences than the species that are not really related. As I briefly mentioned about mutation and sexual reproduction, they are the two processes that produce genetic variation that makes evolution possible. So, in these processes, only the genetic part of variation can have evolutionary consequences. Mutation is a change in the nucleotide sequence of an organism's DNA and the ultimate source of new alleles. In organisms that reproduce sexually, most of the genetic variation in a population concludes from the unique combination of alleles that each individual receives. Natural selection is when there is differences in survival and reproducing among individuals in a population through having interaction with their own environment. So some of these individuals can have alleles that generate traits that enable them to survive better in their living environment. So the more successful an individual will have more offspring. Overreproduction just simply means more offsprings are produced than when they can possibly survive. Phenotypic variation is a variation among population that relate to their behavior or appearances. So this diverse gene species is important for the survival of species in a changing environment. Differential fitness in the environment basically is saying when a organism is living in a different environment, then different genetic variation can be selected. The traits of heritability of fitness can be passed from generation to generation. And in this case, an adaptation is a genetic variation that is selected and is manifested as a trait that provides an advantage for an organism to survive in a particular environment. Natural selection acts on individuals, but its effects occur in the populations. And natural selection acts on phenotypes, but evolution are consisted of changes in allele frequencies. Natural selection does not lead to perfection. Since the natural selection increases the frequencies of alleles that provide reproductive advantage, causes adaptive evolution. The preservation of genetic variation. Because most eukaryotes are diploid, a considerable amount of genetic variation is hidden from selection in the form of recessive alleles. Balancing selection is a selection itself may preserve variation at some location. It occurs when natural selection maintains a lot of forms in a population. Heterozygote advantage occurs when individuals are heterozygous at a particular location, having greater fitness than both kinds of homozygotes. Sickle cell allele is most common in Africa, where malaria is widely spread, as the heterozygote provide protection against malaria. And frequency-dependent selection is when the fitness of a phenotype declines if it becomes too common in the population. The frequency of left mouth scale-eating fish, it changes because the right mouth ones are favored by selection when the left mouth ones are common, and vice versa. Mutual variation is when DNA variation has little or no impact on reproductive success. Sexual selection is a form of natural selection where individuals with inherited characteristics are more likely than other individuals to have their own mates. Sexual selection can result in sexual dimorphism or also marked differences between the two sexes and the secondary sexual characteristics, which are not directly associated with reproduction or survival. These distinctions covers differences in size, color, ornamentation, and behavior. Sexual selection operates in several ways. They are intrasexual selection and intersexual selection. Intrasexual selection is when individuals of one sex compete directly for mates of the opposite sex. In many species, intrasexual selection occurs among males. An example in this picture is a male northern elephant seals fighting fiercely. And intersexual selection is when individuals of one sex, usually the females, are choosing their mates from other sex. The example of the picture is a horned peacock males competing to be chosen by females. A genetic drift is an event happening only by chance which causes allele frequencies to alter unpredictably from one generation to the next generation. So in this picture, this is an example of genetic drift. This shows how genetic drift can affect a small population of wildflowers. So there are two effects in the genetic drift. First one is the founder effect. It occurs when few members of a population migrate to form a separate new population. The next effect is called bottleneck effect. This occurs when a population size severely decreases and allele frequencies of a population dramatically change. There are several more effects of genetic drift. Genetic drift is significant in small populations. It can cause allele frequencies to change at any time, so it's random. It also can lead to a loss of genetic variation within the populations. Genetic drift can cause harmful alleles to become fixed. A gene flow describes the movement of individuals between population resulting in the removal of alleles from a population when they leave or the introduction of alleles when they enter. And the Hardy-Weinberg equation. This can be used to test whether a population is evolving or not. If you take a look at the 5 conditions of Hardy-Weinberg equilibrium, the first one, there must be no mutations, and there's random mating, there's no natural selection, and there's extremely large population size and no gene flow. If you take a look at the Hardy-Weinberg equation, the first equation shown in the slide reveals that allele frequencies for each allele, like the dominant and the recessive, is P and Q. Then the frequency of homozygous dominant is P squared, and the frequency of homozygous recessive is Q squared, whereas the frequency of heterozygote is 2PQ. If we call a speciation, this explains diversity of life, and it not only explains the differences between species but also the similarities between them, also called the unity of life. For the molecular evidence of the unity of life, major features of the genetic code are shared by all modern living systems. DNA and RNA are carriers of genetic information through transcription, translation, and replication. Metabolic pathways are conserved across all currently recognized domains. Second, the cellular evidences of the unity of life in eukaryotes. The structural evidence supports the relatedness of all eukaryotes, such as cytoskeletons, membrane-bound organelles, linear chromosomes, and endomembrane system. Speciation forms a conceptual bridge between microevolution and macroevolution. Microevolution is a change in gene frequencies within the population, whereas macroevolution is major patterns and changes among living organisms over a long period of time. The biological species concept emphasizes reproductive isolation. According to this concept, a species is a group of population whose members have the potential to interbreed in nature and produce a viable, fertile offspring. However, it does not produce viable, fertile offspring with members of other groups. Because biological species are defined in terms of reproductive compatibility, the formation of a new species hinges on reproductive isolation and reproductive barriers. These barriers can be classified according to whether they contribute to reproductive isolation before or after the fertilization. It's formed of prezygote barriers and postzygote barriers. In prezygote barriers, it includes habitat isolation, temporal isolation, behavioral isolation, mechanical isolation, and gamete isolation. Postzygote barriers prevent zygote from developing into a viable fertile adult, and this includes reduced hybrid viability, reduced hybrid fertility, and hybrid breakdown. Speciation can take place with or without geographic separation. Allopatric speciation can occur when two populations of one species become evolutionary change during the period of separation, and it results in the establishment of prezygote or postzygote barriers to reproduction. However, a new species can originate while remaining in a geographically overlapping area with the parent species. This is called the sympatric speciation. It also can result from habitat shifts and sexual selection. This is the end of my presentation, and thank you for listening. Bye!